All right, I wanted to make this a lecture that talks not only about detoxification, but some of the other functions of the smooth ER. It's a really important part of our cells that I think a lot of times gets skipped over because we're really interested in gene expression and protein translation and all of that good stuff. I also wanted to give you kind of a real-life example of why detoxification by the smooth ER is so important, and kind of a link to genetics. So some of this is kind of review that's hit on in other videos. Some of this is more information than you need to know, so you always are responsible for what's in the objectives. But I think this helps kind of make it a little more interesting. So in this chapter we talk a lot about the endomembrane system, and a lot of our focus is on the rough ER because that's where protein translation or some protein translation is happening. So this is really a focus on the smooth ER, and one of the things that you will notice when you look at images is that the smooth ER is drawn differently than the rough ER. But remember that it's a continuous network of membranes. So if a cell needs to do less protein synthesis and more lipid biosynthesis, then the rough ER will decrease in size and the smooth ER will increase. And vice versa. If a lot of protein synthesis needs to be happening and not a lot of other functions of the smooth ER, then the rough ER will kind of be the majority of what you're seeing for that membrane network. And it's kind of interesting. Here's some electron micrographs. Hopefully you recognize these as transmission electron micrographs. And you can see the smooth ER here looks like these long extended membranes with all these little dots, which are the ribosomes. And then as it kind of forms into the smooth ER, you can see that things are looking more like these kind of tubes, and not this elongated membrane. Remember, this is the idea here too is that the more membrane space you have, the more active space, more surface area you have for things to happen. And one of the other reasons that the smooth ER looks more like these tubes coming up when you slice through a cell than the rough ER is because as the smooth ER is making membranes, things are going to pinch off as vesicles and go supply phospholipid bilayers to other parts of the cell. So that's just a little bit of how you could recognize it with the transmission electron microscope. So one of the key functions of the smooth ER in all of our cells, but especially our liver, is detoxification. So you're taking something that could be toxic, could be harmful to your cell, your body, and you're trying to get rid of it. And really what's happening here is we're going to see that detoxification, the big goal is to make a molecule water soluble so that the body can get rid of it. Okay. So xenobiotics means foreign molecules, but this also happens with regular old metabolic waste, which means molecules, byproducts from metabolic reactions that aren't needed in the cell. drugs and alcohol, pretty common, xenobiotics, foreign things, and just any other kind of, organic compounds that your body needs to get rid of. Okay. And we're just really going to focus on this family of proteins called Cytochrome P-450. So Cytochrome P-450 is what we call a superfamily. So large family, multiple enzymes. You'll see it also just abbreviated, like you saw before, as CYP. And there's different CYPs. The 450 stands for the maximum absorbance length for this enzyme. Not something you need to know, but if you're curious as to how it was named. What's amazing, I think, is CYP proteins are found in all kingdoms of life. Right? So that means prokaryotes and eukaryotes. Or maybe I should say eukaryotes and prokaryotes, like we're talking about eukaryotes. There are other metabolizing detoxification enzymes. Like I said, we're just going to focus on the CYP group. There is at least 57 human CYP genes. And then I'm going to show you in a little bit, you can have lots of different alleles for those. So this chart I know is small, but it's just to illustrate to you that CYP enzymes have a diverse set of roles. So they're important in biosynthesis, making things, as well as metabolism, which usually means breaking things down. So you can see CYP2, drug and steroid metabolism. CYP4, some fatty acid metabolism. But then CYP7, you have bile acid biosynthesis. And here's some steroid biosynthesis. So they have different functions in the cell. What we're going to focus on are the ones that are deactivating or detoxifying for this lecture, but there are also some that are important for activating drugs, and we'll touch on this in a minute. So in general, and one of your objectives is to understand this function of cytochrome P-450 enzymes, and I list, or your book talks about monooxygenases and a hydroxylase. Knew it didn't sound right. So remember, if you see the ASE ending, that means it's an enzyme. These both, as shown down here, add an OH group to a molecule. So here to a drug. You know that OH is a polar group, just like OH in water, and the OHs we look for like on carbohydrates that make them polar and water soluble. So if you have a drug and you add this hydroxyl group onto it, it becomes more water soluble. So both monooxygenases and hydroxylases add OH groups to a compound. They just do it by different mechanisms. The other specific enzyme that the book talks about and is in your objectives is the aryl hydrocarbon hydroxylase. You may have seen this written as AHH. So this is telling you what it does. It adds a hydroxyl group. This is telling you what that specific enzyme works on, aryl hydrocarbons. So again, it's adding an OH group, making something more water soluble. But in this case, it's working on something called a polycyclic hydrocarbon. And polycyclic means multiple circular type of molecule structures. So we find polycyclic hydrocarbons in a lot of pollution. And so I just brought up this slide. Cigarette smoke is very well studied for the carcinogens in it. So carcinogen means cancer causing. And though our body tries to add little hydroxyl groups and get rid of these, they still can be quite harmful. So the mechanism we're talking about is for Cytochrome P-450 enzymes is to add an OH group to a molecule to make it more water soluble so that these molecules can leave the body. Now, we're going to revisit the Ames test in Unit 4 with cancer. You may have seen this in general biology or in genetics, depending on your teacher or what you've taken so far. And you don't need to know about the Ames test for this chapter, but I want to introduce it because it's a way to test for something that can be cancer causing. And what's really interesting, I think, is that sometimes our detoxification enzymes actually make something more toxic. So one of the ways that we test for carcinogens using the Ames test is you have your sample, right? Something that you suspect is going to be harmful. Right? And you add to it liver extract. Basically, you are adding the detoxification enzymes. Because what you want to know is, as this compound goes through our body and is reacted or acted on by the cytochrome P-450 and other detoxification enzymes, does it get marked as, and so it will not be carcinogenic, or could it actually be more toxic, right? And so if you didn't have rat liver extract, right, you would just see if that compound on its own was toxic or not. Okay, and we'll talk more about how this all works. You're basically using some mutant bacteria and seeing if your suspected drug, molecule, toxin actually mutates that bacteria back to being able to grow. So the point I want you to understand right now is that sometimes the enzymes in our smoothie R can actually cause something to be a carcinogen when it wasn't before. And that's the whole point of using liver extract in the Ames test, is to look at the effect of our natural metabolism in our body on a molecule and see if it becomes cancer causing, potentially. So keep this in the back of your mind. Again, we'll talk about Ames tests with Unit 4. Okay, so here is what I think the cool stuff is in understanding smooth ER and cytochrome P-450 enzymes. So this is a specific cytochrome P-450. It's cytochrome P-450 2D6. So this is one example of a specific enzyme. And this enzyme is really important in drug metabolism. And drug metabolism can mean two things. It can mean breaking down the drug, right? So the drug is excreted from your body, or it can be activating the drug, and I'll show you an example. So what we have here is this is representing your 2 chromosomes. So remember, you get 1 chromosome from mom and 1 chromosome from dad. And depending on the alleles, which version of that gene you got from mom and which version you got from dad, different people fall into different categories. So pharmacogenomics is using your genetic information to study how drugs will work in your body. So these little symbols up here, stands for an ultra fast or ultra rapid metabolizer, which means you may either have multiple copies of a specific allele, or that's usually what it means. You have multiple copies, so you have lots of this enzyme. And what can happen is you're given the drug and your body just metabolizes it, breaks it down, it's gone. Okay? You're not going to have much response to that drug. Your body is breaking it down faster than the drug can work. On the opposite end of the spectrum, you can be a poor metabolizer, which means that drug stays in your body for a long time, and this can cause adverse effects. So we always want to hit the sweet spot when doctors are prescribing drugs. They want to give you the right dosage so that there's enough to do its work in your body, but not too much to cause any bad side effects, and not too little to not give you the response. So E stands for extensive, I stands for intermediate. They're just kind of classifications of groups of people that they're starting to collect data on, and helping doctors prescribe or recommend the best dosage and sometimes the best medicine. So not all medicines work for everybody the same. And I kind of had this aha moment when I thought about ibuprofen versus acetaminophen, right? So I grew up taking Tylenol. That's the only kind of pain reliever my mom ever had in the cabinet. And as an adult, I would still have these incredible headaches that they wouldn't go away. And so I think someone one time gave me some Advil or, some kind of ibuprofen, and all of a sudden my headache was gone. And it took me a little while to go, oh, so I've been using Tylenol and that doesn't do anything for me, because who knows what my genetics are and the metabolism or the receptors or whatever for Tylenol, but ibuprofen helps those headaches. So you can imagine with drugs and the different alleles you have, and that's shown right here, people respond to drugs differently. So here is just a little chart about the CYP2D6 alleles. And so you can see all these different alleles with the asterisk and the number. And it tells you what that activity is. So some alleles give you normal activity. Some give you decreased activity. Lots of them give you no activity. So part of pharmacogenomics is actually genotyping people to see which alleles they have, and then to correspond that with the drug information that we have, and get you the right dosage for the right response. I'm going to give you one interesting example. And, whoops. That's interesting. Clopidogrel is a gene that helps with blood clots. So clopidogrel is an anti-platelet drug, and its work is to prevent blood clots. You don't want blood clots because that can give you inadequate blood flow to certain organs. So what it's trying to show you is that clopidogrel is taken in, so it's taken in orally, so you're going through the stomach. And then it actually has to go through the liver to become active. So certain CYP genes are important for activating, so for making the clopidogrel work correctly. And so they're showing you an example of genotyping people, so knowing which alleles you have to know what type of metabolizer you are. So in this case, if you're a poor metabolizer, you're never going to get that active drug. If you are ultra rapid, you could have something that gives you too much drug too fast, too much active drug. So this is an example of where a drug has to be activated by our enzyme livers, our liver enzymes. And so it's super important to know what a person's genotype is. So there's information for doctors that says, okay, when you're considering clopidogrel for antiplatelet therapy, look at their genotypes. If they're ultra or extensive metabolizer, you can probably do the standard dose. If they're intermediate or poor, try a different drug. Try something that will give the right, what we call therapeutic window. So to illustrate this even more, if you have a low level, right, so you're not being able to metabolize and activate this drug, okay, on-platelet treatment. Did I say this right? Yeah. Sorry. Let me say this differently. All right. So, if you metabolize the drug too much, no. So what this diagram is trying to show you is that you need to know the genotype so you can get into this therapeutic window, because if the platelet reactivity is too high, which means not enough active drug, you can't prevent those blood clots, you have risk of ischemia, which is inadequate blood supply to your heart or other tissues. If you have too much active drug, you have a bleeding risk because you've lost you've lost the ability to form clots, and you don't want someone bleeding. So this therapeutic window is where we're trying to get with the correct dosing and the correct drug. Okay, so other functions of the smoothie are I don't know, sometimes I like to post goofy science videos. So got the smooth ER, and you got the rough ER. So I will let you read these at your pleasure. Okay, other functions of the smooth ER. We talked about synthesis of steroid hormones. So I just want you to remind you what this means. We don't talk in detail about hormones, different hormones in this class, but we do talk about signal transduction and hormones. So cholesterol, we've talked a lot about, is important in our phospholipid bilayer, our cell membranes. Cholesterol is also a precursor to a lot of these steroid hormones. Remember, a steroid is a lipid, and even though these are big molecules, they can go through the plasma membrane because they are mostly hydrophobic, and they affect gene expression. So that's how things like testosterone and estrogen and stuff physically affect you, is that they turn on certain genes that are going to do things to cells, that are going to do things to tissues. So sex steroids, we know there's a lot of male and female hormones, stress hormones, really important in carbohydrate metabolism. So when stress hormones are stimulated or high levels, they can increase our production of carbohydrates from amino acids, and then they make us store it as glycogen. So we're storing up all this energy in case we, in the olden days, fight or flight kind of reactions. Aldosterone is a steroid hormone that helps regulate your salt-water balance. Therefore, it helps or it affects your blood pressure levels. So all of these are made in the smooth ER. Smooth ER is also very important for the breakdown of glycogen, which is a polysaccharide, into the monosaccharide glucose. And glucose goes out into our blood to give other cells energy. This is really important in the liver cells. So smooth ER is a big player in your liver cells. Another type of smooth ER is found in your muscle cells. And we will talk about this in chapter 14. I think muscle cells are so cool. So this is just a part of a large cell. So you can see all these mitochondria in there for making energy, and the sarcoplasmic reticulum, the smooth ER kind of evolves or is renamed into the sarcoplasmic reticulum, and its purpose is the storage of calcium. And we will learn that when your muscles are stimulated through an action potential to contract, you have to release all of this calcium from the sarcoplasmic reticulum in order for your muscles to contract. Okay, that's a little bit about the smooth ER.